All right, so next we're going to talk about what are known as gene mutations, okay? Before I get into this, hang on. I'm going to go back to this for just a second, okay? So this is where we ended up on the last video. We said that, again, DNA is going to be stuck in the nucleus. We've got to make messenger RNA from DNA through the process of transcription. And then that mRNA is going to be read by the ribosome in the process of translation to build a protein. So again, here's your mRNA being read by the ribosome. According to base pairing rules, the codon in the mRNA will match with the anticodon on the tRNA molecule. If they match, then that's going to send a signal to the cell to drop off the amino acid at the ribosome. The tRNA is going to leave. It can go back into the cytoplasm and pick up another amino acid, okay, of the same type, obviously. And so then the ribosome is going to continue to read the length of the messenger RNA. It's going to do that several times, make several copies of the same polypeptide chain, and then multiple polypeptides get put together to make a protein. So in the grand scheme of things too, mutations can occur in DNA, and that's what we're going to talk about, okay? So any change in DNA or mistake in a DNA sequence is known as a mutation. Some mutations are good, some mutations are bad, some mutations are known as being neutral, okay? So good mutations. We talk about the fact that good mutations are those that can drive the evolutionary process. So more to come on that eventually. When we get to natural selection at the end of the year, then we're going to talk about pulling back to structure of DNA and how we have such diversity that we have on Earth as DNA sequences have changed to drive the process of species changing over time, okay? Bad genetic mutations might lead to a genetic disorder. They might lead to cancerous cell growth or something else that could be seen as bad. Then there's neutral mutations, okay? Neutral mutations we'll talk about in just a minute a little more specifically because they can only be one of the two types that we see here. But when we talk about mutations in general, that term gets a bad rap, okay? But they're not always bad. We are going to focus specifically right now on only two types of mutations, okay? Point mutations and frameshift mutations. Both of these can be totally random, or they may be caused by environmental factors. You've got some examples here like X-rays, chemicals, radioactive substances, UV light. Notice a lot of those things are the same things that can lead to cancerous cell growth, right? And so then if the DNA mutates, okay, if it happens because of some environmental factor, that's often going to be a bad mutation, okay? If it's a random mutation, it's more likely to kind of maybe fall into the good or the neutral category as well, okay? So, two types. One of which is if we have what's known as a point mutation, where this is a change in a single base pair in a DNA sequence. So that's also called a substitution. This is where we see some that are going to be neutral mutations, and they don't impact protein function at all because the amino acid may not change. Right, like leucine, for example, has 6 codons that code for that. So what if it just changes the correct, the nitrogen base that just leads to making a leucine again, okay? Some of these can be really bad though, even though they're just a simple substitution. All right, then we have what's known as a frameshift mutation. And so this is going to be the result of either an addition or deletion of a base pair in a DNA molecule. As a result of that, you're either adding a base pair to a DNA sequence or you're removing a base pair. And so this is always going to be bad because everything that is going to change after that, in the sequence of DNA, if you're reading along the sequence, it's going to change everything after the point of that mutation. Hang on just a second. Okay, so I always use this simple sentence as an example of a point mutation analogy, right? So every word in this sentence, the cat ate the rat, is made of 3 letters, okay? If we change just the A in ate to a C, okay, we maintain a lot of the integrity of that sentence. Of course, that's not a word, CTE, okay? And so it doesn't mean the same thing, but we can still see the same words on either side, okay? When we look here, right, we've got GCA, this is where our change is happening. GCA in DNA would produce CGU through transcription in mRNA. The amino acid is then going to be arginine. When you read your mRNA codon chart, CGU codes for arginine. Well, here we've got GTA instead of GCA. That's going to make CAU during transcription in the messenger RNA. If you read your codon chart, CAU codes for histidine. Okay, that's not a neutral mutation, but it could be a good mutation. It could be a bad mutation. No way to know. Okay, and it's really that there's no way to know unless we have some specific evidence from the organism that we're talking about. Okay, something visual, something physiological that we can see. Okay, with that last mutation. Now we've got a frameshift mutation that can again be either an insertion or a deletion. So same sentence, the cat ate the rat, right? Here we're going to delete the A and 8. Okay, when that happens, then you have to shift your reading frame because we read in sets of 3. So we still have the cat, okay, and then that's going to shift and make this TET, okay, join together. Then we have her and at. And I realize that those are words, but now this sentence is pretty meaningless, okay? And so then the other example is an insertion. So if we insert the letter G in front of the word 8, now we have the cat, gat, F, era, and T. Again, meaningless, okay? So here's our change here. Our original DNA is TAC. That's going to produce AUG in messenger RNA during transcription. You read your codon chart, that's going to make methionine, and then remember that's also going to start protein synthesis, right? So now instead we are inserting a thymine right here, right? When we insert that thymine, that makes this AUA, right, which is going to code for isoleucine, right? So that has now shifted this sequence that we see originally up there and made it this sequence, and we have this extra base that's not going to code for anything. Here, with that example in the sentence, we have 2 bases that aren't going to code for anything. So that's going to shift your reading frame, which means that every single amino acid is going to be different. So it obviously cannot be the same protein at all. And then just real quick, you've also got this slide in your notes. So if this is our original DNA sequence, this is a point mutation, okay, or substitution where we're just changing cytosine to an adenine. Again, could be good, could be bad, could be neutral. Actually, it's not going to be neutral, okay. And then an insertion is being shown here where a thymine is being added between the first 2 bases. So now we have this extra adenine that doesn't code for anything, that shifts the whole reading frame. And then we have a deletion that's cutting out the original guanine, that second base. And so now we've got 2 bases that don't even produce a codon to code for anything. All right, so we will eventually talk about the fact that there are other types of mutations as well called chromosome mutations. I'm not going to cover that here. We're just sticking with these 2 specific types that are going to occur at a specific location, either adding a base, deleting a base, or changing a base in a sequence of DNA. Chromosome mutations are going to involve larger sections of DNA, okay? If you have any questions about this, let me know.